Okay, today we're going to be talking about protein synthesis. So this is essentially why we have DNA. So our objective for today is that you will be able to differentiate between the three types of RNA, transcription and translation, by modeling the process of protein synthesis. And we'll be creating this model on Wednesday and Thursday of this week. And then, of course, we have our mid-assessment quiz, so like a big quiz, this Friday over DNA, replication, transcription, and translation. Okay, so everything we did last week plus everything we're doing this week. All right, so DNA review. So far we've learned a few things. One, we learned what DNA is, right? DNA is a nucleic acid. It stands for deoxyribonucleic acid, and it's the genetic material that contains our genes. Okay, it's really important. DNA contains our genes, which is the information that codes for essentially who we are. Okay, it codes for all the different proteins that give us blue eyes or brown hair, or if we're tall or if we're short. DNA gives us all those directions. All right. We know that in a eukaryotic cell, the DNA is located in which organelle? The nucleus, right? So DNA, at least in eukaryotes, is in the nucleus of our cells. And we learned about how it is replicated in order to prepare for mitosis, right? We talked about replication, the DNA, right? We talked about how it unzips, and then the new bases are base paired on to either side in order to make two new strands, okay? But why do we have DNA? What does it do for us, okay? We know that it contains our genes, and that's really what it does for us. It provides a set of directions, our genes that code for specific proteins to make us who we are. So DNA, we have DNA because it controls the systems of the cell by creating proteins that the cell uses to carry out all of its functions. All right, so if it's going to be a muscle cell or a skin cell or a brain cell or something like that, it needs to carry out specific functions. And the DNA is what codes for all the information for the cell to do those functions. So when we talk about protein synthesis, that's really the whole purpose of DNA. DNA makes proteins in this process, and there are two steps to protein synthesis. The first one is transcription, which you can see in this picture, takes place inside the nucleus. And the second part is translation, which happens in the cytoplasm. So step one of transcription. So here's a picture of what transcription looks like. We already said it takes place in the nucleus. So what are the steps of transcription? The first step is that the DNA is opened up, very similar to replication. Okay, we open up that ladder and we form a messenger with all the information that's on the DNA. Okay, this messenger is made up of RNA. RNA is a single-stranded molecule. It's smaller and it can squeeze out of the nucleus back into the cytoplasm. So the DNA then closes back up and stays in the nucleus to be read again later. Step two is translation. All right, so here's our picture of translation. So the messenger that we just made of RNA, the messenger RNA, goes into the cytoplasm and finds a ribosome. The ribosome helps read the messenger RNA. It matches transfer RNA to the messenger RNA, and by doing so brings in an amino acid that the DNA coded for. Remember, proteins are long strands of amino acids, and proteins are what make up our traits, like eye color and height and everything else. So the amino acids bond together and form a protein, and that's simplified essentially the two steps of protein synthesis. So overall, we say the central dogma or the central concept of how protein synthesis works is that it goes from DNA to RNA to protein. That's how the flow of information goes. So how is the DNA read? The DNA is read and turned into a messenger called mRNA, or messenger RNA. RNA is a single-stranded nucleic acid that can bind to other nucleic acids, but is much smaller than DNA. Some unique features of RNA include the fact that it's single-stranded. It can go into the cytoplasm, right? The DNA is really big, so it's locked up in the nucleus. It cannot get out of the cell to where the ribosomes are. So we need that messenger RNA that's smaller, single-stranded, and can go out into the cytoplasm. Now note that RNA uses a different base. It uses uracil instead of thymine. So we know in DNA the 4 bases are G, C, A, and T, okay? But in RNA, the 4 bases are C, G, A, and U. All right, so it uses uracil instead of thymine. But it uses the same base pairing rules as DNA. C bonds to G and A bonds to U, since there is no T. Okay, so let's practice making some RNA. So let's look at transcription, our first step. Here's our picture of transcription, just like we said. All right, and we're going to read this segment of the DNA to make proteins. So let's look at our DNA. Let's say the DNA, that little spot right here on the picture, okay, is as follows. It's ACC, TTT, CTA, AAG. So the whole point of transcription is to make a piece of mRNA. And in order to make the mRNA, it uses RNA molecules and bonds them to the DNA, using the same base pairing rules that we learned about in replication. So our mRNA strand would read what bonds to A. In RNA, there is no T, we use a U, so it would be U, C bonds to G, C bonds to G, so UGG would be the first part. See if you can figure out the rest before I show you the answer. Okay, hopefully you were able to figure it out. Our RNA, our piece of mRNA, would be UGG, AAA, GAU, UUC, because those are the bases that would bond, right? That whole idea of complementarity, those are the bases that would bond to the DNA. All right, perfect. So now this mRNA piece has the information from the DNA and it goes out into the cytoplasm where it finds a ribosome, right? This is where we start translation. So let's practice translation. Now that we read the message that was sent, here's our mRNA as we created from the DNA strand. And you can see right here that it's all the same letters that you just created on that first mRNA strand, all right? So there's our mRNA. Transfer RNAs, or tRNAs, read this message and transfer the amino acids to the ribosome that is holding the mRNA. So there's a picture of our tRNA. We can see that it reads the mRNA by binding to it. Same base pairing rules apply. And it also brings over the amino acid on the top. So how do they read it? I just gave you a hint. Good, the mRNA is read by base pairing. So the tRNA contain bases. We can see right here these little anticodons that can match up with the mRNA. So this AAG anticodon would be able to bond with the UUC from the mRNA. When that happens, it brings in this amino acid and bonds it to the chain in order to make our protein. All right, so the complementary TNAs bond to the mRNA, and when they do, they attach their amino acid to the growing amino acid chain, just like I said. All right, so looking at our mRNA strand, which we discovered was this, we can tell what the tRNAs will be. Okay, they're going to be the base pairs that would match up with that mRNA. So U bonds to A, G bonds to C, G bonds to C, A bonds to U, U, C, U, A, G. Okay? And these tRNA ones each code for they each are attached to a specific amino acid. And that's how we grow our amino acid chain. So how do we know what amino acid will be added? Well, we can read the mRNA also. And I can see there's a typo. This should say can. I apologize. We can read the mRNA too. So we can use this chart to read the mRNA and figure out what amino acid is going to be brought to us. So for example, let's say our mRNA was AUG. Okay? You read this chart by looking up the first base. Okay? A would be right here. Then we look at the second base, U. Then we look at the third base, G. A, U, G is right here. Now, actually, let me tell you something. We're going to hide this. And we're going to hide this. Because this is labeled incorrectly. All right? So you're going to read this left to right, just like you would read anything else. All right? So here's our AUG. We start here. A. Go to U. A. Look at U. And then we find the G. And we can see that's methionine. All right? And we're going to do lots of practice with this, so don't worry. So, did you notice that all the RNA is read in 3-letter words? Each of these 3-letter words is what we call a codon. All right? So, there's a codon because there's 3 bases. Here's another codon. Every 3 bases is called a codon. So, we can read these codons using the codon chart to determine what amino acid is coming next. So, here is another one. Looking at our mRNA strand from earlier, our amino acids would be block some of this extra information that you don't need to know out. Okay, our amino acids would be UGG. So first we look at U. Then we go to column G. Then we look at G, and we can see that's tryptophan. Okay? So you follow the chart, it's tryptophan. Let's look at the next one, AAA. So you first find the row, then the column, and then the third base. So AAA would code for lysine. All right, and we can see that on the chart right there. How about GAU? What would the base be for GAU? Good, it should be aspartic acid. UUC comes out to phenylalanine. And that is our whole sequence, okay? So we know from our mRNA that our amino acid chain would be tryptophan, lysine, aspartic acid, and then phenylalanine. And that would be the start of our protein. So putting it all together, it's important to realize that DNA codes for proteins. These proteins are made during protein synthesis, which is done in 2 steps. The first one is transcription, which occurs in the nucleus. where the final product of transcription is an mRNA strand. And then step 2 is translation, which occurs on the ribosome, and the final product of translation is a protein. So with that being said, go ahead and take a minute to watch this Amoeba Sisters protein synthesis review video. And after that, you can start the practice worksheet in your EN. It should be a blue worksheet, I believe, and it's going to give you practice using this codon chart so you can get used to it. All right? So each one gives you an example of DNA. You're going to find the mRNA strand and then use this codon chart right here in order to figure out what the amino acid sequence is, okay? Then, once you know the amino acid sequence, you can use the protein chart to figure out which protein it stands for. All right? Give it your best shot on these. Use your notes. Go back and forth. Work with a partner. But try your best, because tomorrow we're going to go over these answers in class, and you'll have some time in class to figure out what you were doing wrong, correct your papers, and make sure we're on the same track before we start actually putting this all together in our foldable. All right? And that's it for protein synthesis.